And the idea of this module here is to teach you how to use compartments so you can make cells of different shapes and have each regions and have each one of those different regions have different properties so you can do more uh, 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 more things, right? The cells can do one thing on one side, one other thing on one side. And one thing that you'll be learn how to do is how to create cells that move, like what you see here in the back, and how to have multiple cells that move, or have epithelium, or have cells with internal compartments, as you see here on the first figure on the left. So remember that this meeting is being recorded and live stream, and that you can feel free to send questions in the chat because it's on Zoom, right? We can now use the Zoom logistics. You can use the chat to communicate. You can click to raise your hand or ask go faster or slower and all these things. Okay, so this is the schedule as of the beginning of the week, right? And now you're on day 6. And this is the first session, compartmental cells. What you're gonna see after the first break, we're gonna talk about how to make links between cells and between, so you can do other kind of morphological processes. So the idea of this module, here's the outline. I'm gonna give some motivation, and then we're gonna go through 2 examples of how this come out, and the last how to build a stable epithelium, and how to create cells, and how you go about making cell compartments, right? You know how to go about make cells, but today is gonna about cells with multiple with internal structures. So what you know is that if you know a little bit about cells, you know that they are spatially heterogeneous, right? The cell is just not a blob that where things a membrane with a bunch of liquid inside where everything is amorphous. It has internal structure, and this internal structure can actually be translated to some external structure. For example, the cells that move, right? The nucleus usually behind. Behind you have information of the stress cables and some contractile events in that cell retracts on the back. On the front, you have the formation of the labellipod and the cell moves. So there is clearly at least 3 compartments here. So we have some or behave. So one behavior on the front where the cells are actually crawling. The cytoplasm stay inert. Maybe on the back there's a little bit of contraction. The nucleus is inside being dragged by this process. So this is the case of a cell that's crawling. So it's polarized and has a front and an end, and you cannot properly model this if your cell model is only made of one compartment or there's no spatial, some kind of spatial heterogeneity to your cell model. Okay. So another case where it is important to take into account that the cells have different functions, different regions of it, is in the case of epithelium. So we have all those cells in epithelium that forms a stable epithelium, and usually have a apical side that faces a lumen or some kind of other liquid, and then and you also have a basal side that usually sits on the next to a extracellular matrix. And those 2 sides of the cell, they have very different uh, properties between another, and this is something that you may want to add in CompCell. The other thing is that is different, that the cell also has a nuclei. Maybe those cells are very squished, and then the nuclei resolves in given the cell shape. You also have the cell have different adhesion properties on its lateral sides to actually couple tightly to its neighboring cells that form the epithelium. So you actually may have different adhesion values between the cells are their size than what you have on the top on the apical side or the bottom on the basal side. So these are all cases where the cells has a clear heterogeneous organization, and its membrane properties vary depending on which side the cell it is, and maybe these internal properties would vary. So these are all cases where probably want to model your cells not as a single compartment cell, but made of multiple compartments. So then you can adjust each one of those properties according to which compartment of the cell you are talking about. Okay, so when you talk about cell compartments, there are two ways to think about it. You can think of using compartments to actually model structural heterogeneity of the cell. So you can have maybe, for example, have a compartment for the membrane of the cell, another one for the cytoplasm, and another one for the nuclei. So here in this case, you're using cell compartments in cell to model actual structures of the cell that you can easily see. So here, membrane, cytoplasm, nucleus maybe also add the Golgi or the mitochondria, wherever you think is really useful for your cells, you can add to that. Another way that you can do is actually use compartments to model a more functional regions of your cell. Maybe, for example, in the case of the apical cells on epithelium, you don't have an apical structure and a basal structure and a lateral structure. What you have is an apical region of the cell that has different properties than the basal region of the cell that has different properties. And then you can play. So in this case, you can make your cells made of three compartments. They do not directly correspond to any physical structure of the cell, but they respond to some kind of gradient or where well, ultimately, you have different proteins on different regions, right? But you cannot single point to a specific protein or a specific organelle inside the cell that sets what's the apical, what's the basal. So in this case, you model your cells with different compartments. Each compartment represents a region of the cell. We have collectively a different properties than the other parts. So in the case of epithelium, we may have an apical and basal compartment. You maybe have three compartments, an apical, basal, and lateral. You may even add the nuclei inside, so you can actually mix some concepts like structural compartmentalization um, with conceptual compartmentalization of a cell. So these are things that you can do, and then it comes up to you as the modeler, the model builder, to choose how you want to go about it. So the first published paper that used compartments in the cellular pods model was this a paper by Starus, where they model myxobacteria, and because it's very long bacteria, you cannot properly model that with just one cell compartment. What they did, they model the bacteria as made of multiple compartments. They are all exactly the same from another, with the exception of the two on the edges. And I think one of them is the one that is the head, and then the ones that crawls, and the other ones just follow behind because you have some kind of coupling between those compartments. Okay, so that is a case of kind of a mixture of structural and conceptual compartments of your cell, where there's a clear difference in behavior between those two on the edge and the one in the middle, but otherwise, this subdivision here is more just for modeling purposes to make things easier. You can also do that for the epithelium I just talked about. And one thing that you can do, you can, on top of that, add different behaviors of different regions of epithelium. For example, you can add spring forces between two compartments that are neighboring. This is something that you'll be learning how to do a little bit today, but you're going to do for this specific example, you're going to be learning tomorrow. And then if these cells exert those forces, then you have constriction of one the size of your epithelium. If at the same time, for example, this model, you make this other side of the cell to grow, then you can actually induce folding and invagination of your epithelium. Okay, so this was done by Dr. Abbas Shinifar. Okay, so, and of course, there are many other applications of use cell compartments. Once you have know how to do cell compartments, you can do more more complex structures like in this simulation here of the renal cyst, uh, the renal tube, where we actually have where the cells have a apical side, the basal side, the internal side here, and you can change the properties of the lateral side that makes the connection between other cells. So you make one cell to have a little weaker connection, you can pop out from the tube and makes a cyst outside. And so this is an example of using this. And the other thing that you can use, you can also make those compartments, instead of making them static, you can make them dynamic. And by dynamic, I mean that the cells can mix inside the cell and sort out according to the stimuli from the environment. So that's what we call dynamic compartments. And with this, you can model things like PCP signaling, which is something that we'll be learning how to do on a specific module tomorrow. Okay. Right. So this is overview of how uh, cell compartments work in Compartmental 3D, right? And you may ask yourself, okay, having already covered cell compartments in this course, because you probably have seen this movie and learned how to make a simulation of a cell that crawls, right? Which has a nuclei, a cytoplasm, and a lamellipodia, right? Have you guys seen this? I think you've seen that in 4.1.2. So you may say, okay, I already know how to do this. Why do I need a specific module for this? Well, the answer is that not really, because if you just pick up that model and now you put two cells together, right, and you play, you may see that at beginning everything looks fine, okay? You have the three cell types, and they are behaving as the way they should, but then something bad happens when they meet each other. Is that the problem here, and also is even worse when you add more cells, is that once they meet each other, the nuclei doesn't know if one should belong to this cytoplasm or that cytoplasm. Is the lamellipodia belong to this cell or that cell? So once those cells meet together, they just mix, right? There is no information about telling, okay, these three compartments belong to this cell, and these other three compartments belong to this other cell, okay? So you can solve this by generating a new set of cell types. So you can have nuclei 1, cytoplasm 1, lamellipodia 1, and then three more cell types, nuclei 2, cytoplasm 2, lamellipodia 2. And then if you want to three cells, you can add more three cell types, right? But then if you want to model multiple cells or a system that grow, you cannot really do this, right? So there must be a smarter way to do this. And there is a smarter way and there's a proper way. And the idea here is that you subdivide your cell. You give another information to your cells, which tell, okay, this set of regions here belong to cell 1, and this set of regions here belong to the cell 2, right? And the way you do this is you assign a different number to the cells, okay, that tells them, okay, these are cells 1, all these compartments belong to cell 1, and all these compartments to cell 2, because they have this different flag called the cluster ID. And when you do this, you can set different contact energies between your domains. One between cells that belong to the same cluster, and other between the cells or domains that belong to different clusters. So you split your contact energy into two. One would be the external contact energies between domains of your cell that belongs to different clusters, and an internal contact energy, which sets the energies of contact between domains that belong to the same cell. Okay. So I just said that. So if you want to do this for the cell crawling example. This is where we use the original values from module 4.1.2, sorry, the number is wrong here. So we set 11, 10, and 40 as the values, and that's where it goes. And if you do this, the cell will behave as we have seen 2 days ago in the example of cell crawling. Okay. And then for the contact energies, if your cell only has one copy of its domain, so the cell usually just have one nuclei, one cytoplasm, and one lamellipodium. Okay, then there is no cytoplasm energy, no lamellipodium energy, and no nuclear energy because your cells only have one of these compartments. So you can ignore this diagonal here. Okay. And then the external contact energies, which is going to be your standard contact energy that you have in a CompCell 3D syntax, is the contact of all its compartments with whatever everything that's external. So all the counts of them with the medium, we can still use the old values that we use for that example. So in that case was 22, 10, and 100, okay? And for these middle ones here, we have to kind of reason out, okay, what should be the values that we use between cytoplasm of one cell and the cytoplasm of the other cell, or what's going to be the contact energy that we should use for the nuclei of this cell with the nuclei of this cell, or the cytoplasm of the neighboring cell that does not belong to it. Okay, so once then, what you have to do now is to make guesses. Okay, educate the guess of what they should be. So how you can reason about, okay, so the nuclei of one cell should never want to be in contact with the other cell, with domains that belong to another cell. So it should never come in contact with another nuclei, never should come in contact with the cytoplasm of the neighboring cell, and should never come in contact with the lamellipodia of the next cell, right? So because you don't want this to happen ever, we can say that the contact energy, the external contact energy of the nuclei of one cell with all the compartments of a different cluster of cells should be very high. So that will prevent the nuclei from ever merging with the neighboring cell, and you keep it inside the cell here. So just like what we did for the contact energy of the nucleus with the external medium, we do the same thing with all the other structures that belong to different cells. Avishek has a question. Please go ahead.
Yes. So I understand that the nuclei and other things should not kind of like walls, but the boundaries of the two cells can touch each other, right? So if I set those values high, then is
The
it
cell
just saying like, yeah,
yeah,
just kind of like this, yeah. They should not overlap. I get that, but they should be doing this. Yeah.
that will depend exactly on how your cells behave, right? So do your cells are kind of refractory with one another? So they once they divide or they accept different, they never touch. Then you make the adhesion between the cytoplasm of one cell and the energy of the cytoplasm of one cell and cytoplasm of the other cell to be also high. So they always have a little bit space between them. If your cells should couple, right, then you can make this energy lower. In this case, the cells actually always be close together and move as a couple, right?
Okay. So in the case
Or
when
if,
they are just,
yeah,
sorry.
and if the cell should follow one another, maybe you want to make the lamellipodia of one cell to really like the cytoplasm of the other cell. So then they will, well, in the case of two cells that work and they make some kind of line, okay?
Okay.
But then you make the cytoplasm of the two cells to not like each other, so they only couple at one to the edge of the other. Okay, of course, the cell can also rotate and kind of couple like this, but then uh, then the specific choice for those energies have to reflect what your particular system. Okay,
Okay. Okay. Thank
makes
you.
sense?
Yeah. Great.
Okay, that's a great question. And we can, so you have to think, okay, how exactly my cells behave, or what kind of cells I want to model, and based on that, you chose those energies. So the first thing you have to do is, I'll say, is to first just choose these energies qualitatively. What should be high, what should be low with one another. So you first make a qualitative table of how the energy should be, and then after that you start adding the numbers, okay, and then adjust. But you should have first this qualitative idea of how the energy should be, right? So you have to reason out. So what I want to stress in this module is, okay, think about qualitatively what should be instead of just plugging numbers right away. Because if you just plug numbers right away, it will take more time for it to make things work as you want. Okay. But that's a very good question. And then, okay, of course, is my next slide. Oops. Okay. So in the case that I want myself to not really mix one another, but kind of move away as independent entities, I usually choose those to be high, okay? But not as high as the other ones. So they can actually eventually kind of touch each other, but they're going to separate right away, okay? If until they touch and couple, then maybe you make this cytoplasm to be lower than before. Okay, so we just discuss about it. So if you make a proper choice of this, okay, then you can have something that looks like this, where you have multiple cells, right? In this case, the cells are dividing, but you see that when they get close to one another, they maybe actually touch a little bit, but they don't really merge. They actually, there's always a little bit of space in between them, okay? So with this, let's go to our first exercise, which will be to create simulation with many moving cells using the cell compartment features in Compartmental 3D. Okay, so in order to do this, first thing you need to do, you need to use the contact internal plugin. You need to specify what the initial compartments belong, that the initial compartments belong to the same cluster. We have to implement cluster division, and we have to choose appropriate contact and contact internal values, okay? So we're not gonna do this all at once. We're gonna break this into small parts, okay? So first, I'm gonna show you how you use a contact internal plugin. Okay, so let's now go about making those things. So the first thing you want to do, okay, is you don't want to code everything from scratch. Let's just use the code that you has developed with Gilberto for module 4.1, okay? So I want you guys to all go to the student materials and download a new version of it, which is stored on 6.1.1a.zip. So please go there and download this file and let me know when you're done. Download your computer, open it and tweet it. And once everybody open tweet, you can proceed to the next. Yes, once you open the zip file, it's called e4122.cc3d because it's from the old module. Oh, what's the error, Sharon?
The error is lamel is not defined.
Lamel is not defined. Okay, let me check. I'll open here on my computer and make sure and check if it works.
The error is it says lamel instead of self.lamel in the Python on line 15.
Once you fix that, everything works fine. So I just uploaded a working version of the code on the student materials. If you already downloaded, the only thing you have to do to fix is on line 15. Instead of having lamel, you have to have self.lamel, okay? If you have not downloaded yet, you can just go to the materials and download it, and now it should work. All right, so let's continue. Okay, so if you now download it and it's working, okay, I'll ask you to open the simulation on 20++, okay, and take a quick look to see how the code looks like. And yeah, probably you guys already did. You certify that it runs as expected. So this is, I guess, is done. So once you have the it loaded and tweeted, let's proceed to the next step, which is I want you two guys to go to line 53 of the code. And on line 53, I want you to add the content internal plugin, which you're going to set the content energies of the internal compartments of the cell. So if you go to, you are on the XML file, right? Go to line 53. There should be a kind of empty line between the contact energy and the uniform initializer. And place your cursor there. And then you go to CC3D ML, plugins, and select contact internal. And then if once you do that, this should put the contact internal plugin for you inside, and it automatically knows which cell types you have in your simulation, and you add everything there in the correct way with the correct syntax for you. So if you do that, you should get something that looks like this, okay? And do not worry about the parameter values right now. You change them later because Compartmental does not know what you want to do. So you just give all the energies to be 10 and the neighboring order to be 1. So these are automatic values which will be changing. It's our job to change those numbers according to the what we want our model to be, how our model should behave. So did anyone get to here? If so, please send a message. If you did not get it, also send us a message so we know where you are.
So that plugin should not be indented.
Oh, for the XML, the indentation doesn't matter. Okay, you probably got something that is right at the edge, like you see here.
Okay.
So XML, it doesn't matter. Python, everything should be nicely indented, yes. But XML doesn't. I still like to ident everything, so you can quickly see the structure with your eyes.
Oh.
But yeah, so you can make an indentation here, this here, and bring all those inside. That will look nicer.
Oh,
Yes.
okay.
So, did everybody got this? Okay, Sharon got it. So I hope Jupiter got it. All right, many people got it. Excellent. All right, so let's proceed to the next step. Okay. Now that we have our contact energy, we need to specify what the initial compartments belong to the same cell, right? So the cell has 3 compartments: nuclei, cyto, and lamel, right? And you want to now tell the cell that they actually belong to the same cluster of cells. So this, let me just tell you how this works, okay? Is that in CompCell or the cell pulse model, every cell has a number, right? So ID. So if you look at any simulation, for example, this simulation here of the somatogenesis, right? You see that every different cell color here correspond to a different cell type, right? But every thing here inside, every different boundary here correspond to a cell. And so here we have many cells that are of the type green, right? And how do you know that they are different cells and not the same cell? Is because each one of those lattice sites has a number associated with it, and this number is 1. All those lattice sites here has a number that which is equal to 4. All these lattice sites has a number that's equal to 3. Okay, so that is the ID of the cell. So the cell has this is the cell with ID 1, this is the cell with ID 4, this is the cell with ID 3, this is the cell with ID 2, 5, 6. Okay, and in your simulation of the crawling cell, each one of those regions will also have a cell ID. Let's say this 24, 32, 107. Okay, I just pick up random numbers. Okay, they do not need to be in any sort of sequence. Okay, so when you have two cells next to one another, right? You need another number to specify that, okay, these three domains belong to one cell, and these other three domains belong to another cell, right? So how do you say that these three domains specify to the cell outline in red, and how do you specify those three domains belongs to the cell outline in orange? You do this by assigning another ID to the cells, which is going to be the cluster ID. Okay, so that cell that you had seen before has maybe ID 24, 32, and 43, but all those three regions has a cluster ID equals to 2, and that tells them that they all belong to the same cell. And now the next cell, the neighboring cell, it has each one of the compartments has a different ID number, 59, 88, 85. But in addition to that, they also have a cluster ID number, which in this case is set to 10. So because they have different cluster IDs, these ones with these ones, you know that this belongs to the cell on the bottom and this belongs to the cell on the top. Okay. So this is another number that you actually can retrieve at the Python level, and you can also assign in the Python level. For example, if you want now to make this nuclei to belong to this cell here, you can change its cluster ID from 10 to 2, and then this nuclei will probably kind of jump from this cell to this one because it doesn't want to stay inside the cell. So while the cell ID is a number that can never be changed, because once you assign that to a cell, you stay with that forever, the cluster ID is something that's more dynamic, that you actually can change either to determine which cell, which compartments belong to the same cluster, or to maybe have some part of the cell to decouple from it or join another. So not sure when this will be used for any type of model, but it's something that you can do. You have freedom to specify the cluster ID to be whatever number you want. Okay, you can change it that mid simulation. Okay. So internal count always comes along with cluster ID. Yes. So by default, all cells have an ID and a cluster ID, but they are set to be the same number. Okay. But once you start using contact internal energies, then the cluster ID actually has a meaning. And if you want to, you can modify the cluster IDs so that the cells, to inform the simulation that those three cells or those three compartments belong to the same cluster, right? Okay, more questions about cluster IDs? So that's the idea. So we need a new number to specify which compartments belong to one cell, to one cluster of cells, and a cluster of compartments, and which compartments belong to another cluster. So that's the idea. Okay, so so I specify how that's done theoretically, right? So we actually have to do this. Okay, so how do you modify the cluster ID of the cells? Is the following. You have to do this at the Python level, okay? So you have to all go to the Python steppables part of your code. So please click on this file here, e4 122 steppables.py, and online 12, on lines 12 to 20, that's where you're gonna do the assignment. So the way we're gonna do this is the following. You're gonna the first thing you have to do, you have to choose one compartment and retrieve the cluster ID of this compartment, and then assign the same cluster ID to the other two compartments. Okay. So the way you do this, you go over the, well, because we start with one cell, okay, this works. So you're gonna go to the first loop with over that grabs the cytoplasm of the cell, okay, and there you're gonna retrieve what is the cluster ID of that cell. So the way you do it is you can make a new line after the line 14, okay? So go here and make a new line, and on that new line, you're gonna use the drop-down menu to grab the cluster ID of the cell. And the way you do is you go to the cell at Compartmental to the Python, cell attributes, and then you have the cluster ID. You click that, okay? Compartmental to the Python, cell attributes, cluster ID. Make sure you made a new line after 14, okay? And once you do that, you have this new line here which says cluster ID. So you just create a new variable, and that's going to be equal to the cell.cluster ID. Okay, so cell.cluster ID gives back the cluster ID of the cell, just like cell.id gives back the ID of the cell. So once you retrieve the cluster ID of the cell, now you can go to the other two loops and assign that cluster ID to those cells. Okay. Again, this code only works because you have just one region of each type. Okay, if you do this, if you have more than one cell, you probably want to do. Okay, good question. Then you have to have some kind of. You see how you can do that in the next example where you actually make an epithelium. Then you see it, okay? But then you need to get more information to know when the cell belongs to this or to the other, okay? Now, okay, so for the next one, you do the cluster ID reassignment, okay? So let me just go there. So first thing, you go there to line 18 now and make a new line, okay? And with the cursor there, go to Compartmental 3D Python, cell attributes, and then now we have something called cluster ID reassignment, okay? So you want to do that for the two other cells types, okay? And once you click this, you get a new line of code, which will be self.reassign cluster ID. And the first entry here is the cell that you're reassigning. So you already have this from this loop from self-lamel, okay? And then the second entry, the second argument of this function will be the cluster ID that we retrieved before. So what this is gonna do, this is gonna tell, okay, now the cluster ID of this cell here will be the same as the cluster ID of this other one. Okay, and you do this for the lamellipodia and also to the nucleus. Okay. So first you retrieve from the cytoplasm, and then you have the cluster ID of cytoplasm, and you assign it to the lamellipodia and the nucleus. And now all those three compartments will belong to the same cluster. Hope this makes sense. So please implement this, save, and try to see. Ali, no, that if you invert that, it will not work. Okay, so the question was, what if I may add this to this side and this to the other side? Ah, as in here? Ah. That's a good question. I never tried to do this. So how about you do the following? You try this one, see how it looks like, and then you try the other one and see how it looks like. I don't think it will work if we try to do directly assign. Doesn't work. Okay, someone tried. Oh, it explicitly say that you cannot do it. Okay. All right. Okay, so that you're right. So you cannot do it from the Python level. We have to use this specific function. So please implement this. Okay, save. Remember, you have to save your code. Okay, so if there's any kind of red symbol here, is because your code has not been saved. Save everything, so everything becomes blue, and then you can try to play that on Compsel. So, sorry, try to do this, okay? And please save and play and see if you got something that looks like this. So when I do save and play, I get something that looks like this, which is not quite what we expected, okay? So in my case, it didn't seem to work. How does it look for you? You guys got the same as the video? Right. Yes. You should be got some that looks the same as the video. Okay. So probably there's another step that you have to do. What do you need to do? What's missing? So why the cell now looks different? Yes, we need to set the contact energies. Exactly. So we create the contact energies a plugin right there. We assign the contact energies, but we forgot to change the contact energies to make the cell look right. Okay. So what we had before is that when you just have the cells as not make a differentiation between them being a cluster or not, we just have this contact energy and we're just use the external contact energies for them to interact with one another. Okay. But now we're told that those cells are part of the same cluster. And when you just add the plugin, you just add the same value to everyone, you add 10 to everyone, okay? So what you're seeing is what happens if all those values are 10, okay? So we do not you want to use this value. What you want to do is now use these values here and add now as the internal contact energies, okay? So we have now to our internal contact energy need to reflect what the old external contact energy was, okay, for at least those three values. So what you have to do now, we have to go to the code at the XML level and make sure that the additional energy between cytoplasm and millipore is 11 instead of 10, and the contact energy between nucleus and millipore is 40 instead of 10. Okay, so please go out to the code and make those changes. And also the other thing you have to do, you have to make the neighboring order from 1 to 4. So please do those changes, and then let me know if now the cell looks right. So Anderson, you have some kind of indentation error, so make sure that on your Python level, the indentation is respected here. So this should be the same level as this one, and those two new comments should be the same level as the previous comments. Okay. It might just be one line, one space, or Yeah, so make sure that it's as you see here in this example. So it seems that Jupiter got it to look right now. Don't know how to pronounce your name. Wisha. I also got it right. Yes, you do have to change the neighboring order from 1 to 4, okay, to be exactly as it was in the external. Kishore got it. Excellent. Okay. So I'm gonna move this. So this is how it should look like. So yes, good answer, Yering. Is correctly right. If you got a 1 on the drop down menu, you have to change this to 4. So the energies have the same weight in all cases, and they have the same reach. Okay. So does everyone now have Kishore, you have a question? Please go ahead.
Yeah, so since we are defining cluster IDs now and all three cells in our simulation have the same cluster ID, the contact plugin, does it become obsolete because is it defining contacts across clusters?
Well, if you just have one cluster of cells, it becomes a little bit obsolete, yes. It still have to not totally, because medium will always be an external entity, okay? You cannot assign a medium to the same cluster ID of your cells. So at least all the values of your cell types with the medium are still there, okay? But as soon as you put more cells, then you need the external contact energy, the other values of the external contact again. Make sense?
Okay.
Yeah.
Yeah.
So still needed for the contact with the medium, right? But things get more interesting if you have multiple cells.
Okay.
And then you will be using that again now. Good question. Okay, can anyone help Susan? Okay.
I have more lines inside of the contact internal than you have.
You have more lines?
Yeah.
Yeah,
Awesome.
actually, I was going to ask about that as well.
Which
Because
Sorry.
the code snippet creates medium energies as well.
the code adds medium.
Okay.
You can certainly delete these ones. Okay. It should not have add the medium as an internal contact energy. So if it's adding, you can just delete it. Okay. All right. Okay. So hopefully you have now a cell that looks like this, and I guess most of you do. So what you want to do now is have cell division. So you have multiple cells, right? So now, once you have multiple cells, then you have to go back and revisit external contact energies to make sure that they behave correctly, okay? So so probably you have already seen, no, I know that you have already seen how to divide cells, right? The question is now, okay, how do we divide cluster of cells? Do you have to divide each one sequentially, right? Or is there a smarter way to do it? Luckily, Compartmental 3D has a smarter way to do it. So let's see how division of cluster of cells work in Compartmental 3D. So for this one, let's go to the to our code, implement cluster division. For this thing, we need a new steppable. So please, in our file menu on the left, right-click on the steppables file. You should open a drop-down menu, and let's go to Add Steppable. Okay. So this is gonna add a new steppable to your code. You'll see, Kishore, how it works. Okay. So add steppables. So you go here, click on this one. And the next, when you click this, you get a wizard menu. And you have to give a steppable name. Let's give it divide cluster cell or just divide cluster. Okay. And on the steppable type, the default is the generic, but you don't want the generic. What you want is the cluster mitosis. Okay. The name of the stable is free, but you have to give something that makes sense. You can read back your code, or your code to be understandable to other people. So let's just call it divide cluster cell, okay? And let's select cluster mitosis. No, visualization fields, in this case, something else. Generic, when you choose mitosis and cluster mitosis, you already come with the code to do the mitosis for a single cell, or we already come with the code to do the cluster mitosis for a cluster of cells. So it's more convenient, you don't have to type everything. Okay, yeah. So select cluster mitosis, give the name, and hit OK. And then you should see something that looks like this, okay? And you'll probably be around line 40, okay? You should see this new steppable, add to your code, and you see that there is a lot of code there that's gonna implement the cluster mitosis for you, okay? You just have now to change to make the division happen the way you want it to happen, okay? So what did you got here? What Compcell gave you is a kind of more or less okay, the code, and there's more about 3 sections. So the first part here, you don't really need it to implement the cluster mitosis, just to show how you can handle cluster at the Python level. For example, for cell in your list of cells, okay, you can, for any cell that you got from simulation, you can retrieve the cluster of cells that it belongs to. So that cluster of cells, you give the cell cluster ID, okay? And you get a list of all the clusters, all the compartments inside that have the same cluster ID, or all the cells that belong to the same cluster, okay? And once you have this list of all the cluster cells with the same cluster ID, okay, all the compartments that belong to the same cluster, you can do a loop of cell local in cell cluster list, and then you can iterate over all the compartments inside your cluster, okay? So this is just to show you how it works. Okay, okay, so this is just to show how you can handle cluster cells, how to can make loops over them, okay? What I highlighted in red is actually where we decide which cells or which cluster you divide. Okay, so first you have a mitosis cluster list, which is empty. And then what you do, you go for compartment list in self of clusters list. So this is a function that returns all the clusters of your that exist in your simulation, okay? And so now you're going to be iterating over all the different clusters. So for the first cluster iterate, you set the cluster ID to be volume to be 0, okay? And then this compartment lists the list of all the compartments inside your cluster, and you can do a second loop of the cell in this compartment list, okay? And then what you do, you store what's going to be the total volume of the total cluster. So you are going over each one of the clusters inside each one of the compartments inside your cluster, and you summing up to this variable, so you know the total volume of the cluster. And usually the way we implement cell division or cluster division in this case is that if your cluster is growing, right, the compartments inside the cluster are growing and reach a threshold, then that's when you want to divide your cluster. Good question. If you do this, don't we need the cluster to grow? Yes, and we don't have the cluster to grow, right? And actually, I don't want to waste time making each one of the clusters to grow. So instead of using this condition, you're going to just use another one. You're going to just flip a coin and decide that the cluster will grow, divide, okay? The code add things like this because usually you want to divide, you want first to make things grow and then divide. But for the purpose of this tutorial here, we want to skip that and we're going to replace this by just a random number. Make sense, Aaron? Okay, so you get there. So here is just how we decide when they will divide, okay? And once we decide they'll satisfy the division condition, whatever that might be, we append that cluster ID to the mitosis cluster list. Once we decide all the clusters will divide, you go and do a last loop over all the clusters that will divide, and we just said, okay, now divide this cluster. Then when you divide cluster, you still have the same options that you have for dividing single cells. We can divide a random orientation, a given orientation, along the major axis or along the minor axis. And they're going to be the major and the minor axis of the whole cluster, not of any individual cell inside the cluster. There is no way to specify a target volume for a cluster of cells. You have to specify a target volume for each one of the individual compartments, okay? You have to make the cycle grow, the nuclei grow, and the lamellipodia to grow. You have to do then that separated. So that is how the structure of the code that you just inherit from the addition of the steppable. I will already talk about this. And here, so circle back to the question from Yaren, okay? We don't want to make the decision to grow to divide based on the cell that's growing. You don't want to implement that. What you want to do, you just let's replace this condition with just a random number. So on the top of the file, please add from random, import everything so you can have access to the random functions from Python, okay? And once you add this line on the top, please replace this with random lower than 0.005, so low number, so we have cell divisions. So please make these 2 line modifications, okay? Replace the cluster volume higher than 250 with random less than 0.005. So that's how it looks like. Okay, I still have here a different number, but it should not make much of a difference. Okay, so for you go over all the clusters and you have a 0.01%, 0.1% or 0.05% chance of dividing at any given time, okay? And the way that it happens is that Compartmental automatically produce choose the division plane for you randomly because you ask for random orientation, and then Compartmental 3D automatically reproduce the compartment arrangement from your original cell into the other cell. So you make this line here, okay? It knows that there is a green compartment on top, cytosol compartment in the middle, and a nucleus compartment at the bottom. So we're gonna reproduce this on two other cells, green, purple, yellow, green, purple, yellow, okay? Will look funny right after division, but as soon as they divide, the cells will sense the contact energies and gonna move away, reestablish their shape very quickly. Okay. So that's what's going to do. The last thing you have to do is that because the cluster mitosis usually assumes that you first grow your cell and then you divide it. It already on the update attributes function, it automatically divides our compartment's target value by 2. We don't want to do this now because we're not dividing based on growth, we're just dividing based on chance. So we can take those 2 lines here and remove them, okay? So we go to the self-update attributes and take this out and let it like here. So once you do this, you can save your simulation and play it. Yeah, and make sure that your simulation was saved before you hit play. If it still does not divide, then you may try to increase the chance of dividing. So please do that, and once you divide yourselves, let me know if you see division, and let me know if after division the cells were correct already or not. So Ali, did you get your cells to divide?
Well, I got it to divide, but the lamellipodium is getting detached because most probably because of the contact energy.
Yes. Yeah. So okay. Yeah, and what you can. So yeah, that's correct. If you run simulation now, things are gonna look weird. Okay. So what you have to do next is set the contact energies, the external contact energies of cells to behave correctly, right? I think we still left the external contact energies to be the same as before. So all the cells still want to belong to they behave in a way that they want to be together, okay? So we have to set this up. Try to set the probability division to be higher so we can see it more quickly. Okay, Kyusha, if your error of run is not defined, please go to the top of your simulation and add the line from random import asterisk star.
I think they will try it.
You have tried? Well, stop simulation, go to edit, make sure that the line is there, save it, and open again in the player. Problem solved, excellent. Okay, so the reason that we got our cells are divided, so that means that you successfully implement the cluster division, okay, but the cells still look weird because we did not fix the addition energies, okay? So what you have to do now is choose appropriate external contact energies. So they're gonna actually look like this and not look like that, right? So what you have to do, you have to go back to the XML file, and we have already did this exercise. What you have to do is we already reason out that the contact of the nuclei with everything else should be very high, and that the contact with the cytoplasm, lamel, and lamel should be also a high number, okay? So what you can do now, you have to go to the player, go to the XML and make a choice of what those numbers should be, okay? So you can either make your own choice now or just copy the values that I have here. And once you do this is a simulation that shows how it looks like when you choose 100 for this line and 50 for those, okay? But if you want to do something else, like Abhishek was telling us at the beginning, maybe you want the cell to couple with one another, so maybe you can reduce this to be a lower value, or maybe you can reduce the cytoplasm, the lamella of different cells to be a lower value, so they will in attempt to want to follow each other, okay? So now it's up to you to choose those contact energies and get the behavior that you want to for your system. Okay. So let me know if it works. We're gonna give you 1 minute to try that, and then we can proceed to the next exercise. Excellent, Jupiter got it. Okay. Now that you got it, you can maybe try different values here and see how the simulation looks like. Yes, Ramon, someone asking is, no, it's a very appropriate question. That, yeah, the difference between contact and contact internal is that contact internal only considers the cells that have the same cluster ID, right? So you want the nuclei to stay inside the cytoplasm of yours, if they belong to the same cluster, right? But you want your nuclei to abhor everything else. So you have a low value of nuclei and cytoplasm in the contact internal, but you have a high value of nuclei and everything else in the external or contact energy. So that's how you play of the arrangement of compartments within the cell with respect to the same compartments with the external cells. So if it's very crowded, you might maybe lower the additional energies of the compartments with the medium, okay, the contact energy. That should help a little bit.
I don't see the nucleus in some of my cells.
Okay, maybe you are dividing too fast, so then the nucleus may be lost. They probably were lost during the division. This is not the proper way to divide, because you usually want the cells to grow first and then you do the division, right? It might happen that you have two divisions right away, and then this means that you divide the cell didn't have time to grow, and then you divide again, and then you may lose one compartment in the process. But the point of the exercise was just to learn how to set the contact energies and the external contact energies, and know how you can divide cluster of cells, because the code for division cluster of cells is different than the code to divide a single cell. So as long as that work, this is already a successful exercise. You can delete the medium terms in the internal contact energy, but not the external. The external, they should be there. Okay, Kishore now got a confluent issue. Yeah, and I see that some cells lose the nuclei. Yeah, once you lose the nuclei, there is no way to put it back, unless you do coding from the Python level. Jupiter, you have a question?
Yeah. So I noticed that when the clusters are dividing, because the model that we worked here is for the directionality to be random, right? To set to random and follow some path. I noticed that the daughter cells have the same direction as the mother cell, so I assume that these attributes get passed on. So I
Yeah.
was wondering, like, oh, Oh, now that I'm talking out loud, I think we already had that on steppables, right? That was the cluster division. And then every attribute from the parent just goes to the child, basically.
Yes,
Okay, cool. Yeah,
initially,
just
right?
speaking out loud, I answered myself, but yes, thank you.
Yeah, but you see that the next time they choose the orientation, there's gonna be random, right?
Yeah, yeah,
After division, they keep going same side, then they
exactly. Awesome. Thank you.
sure.
I'd like to clarify one thing. So is this the contact on the left-hand side of the screen is for the contact between the clusters, right?
Yes, between the
And between the
different between
yes.
compartments that belong to different clusters.
Okay. And the internal is like within the cell.
Yes, within the cluster.
Okay. Okay. Thank you.
The reason you guys are losing the nuclei is because the division process is it's artificial, right? One thing that you can try to do is to make this better is maybe instead of having a dividing at the random orientation, may choose divide along the minor axis. So you're going to divide like this, then you're less likely to lose a cell type in the division. So use this one here. So comment this one and use this, and that might help with the losing nuclei issue.
But I'm confused about one thing. Like, even if it is a random orientation, I'm dividing along the random orientation. Like the daughter cell should get the exact copy of everything the parent cell has, right?
Yes,
So why is it the case that in a random, if I tell it to divide in a random orientation, I can lose the nucleus?
because it's hard to make a precise copy of the arrangements to both sides. So, for example, if you look here, you see that this nuclei got a little bit smaller than this one, just by spatial considerations. Okay, so and because everything got divided, there is a high pressure to everything to grow fast, and the nuclei really dislikes the medium. Okay, so you want to minimize this. This one wants to grow really fast. So this kind of, and because it's a stochastic model, the fluctuations might erase one of the compartments accidentally.
Okay.
So that's why I believe if you just tell to always divide like this, you may get better results.
I see.
All right. So I hope you're having fun with the dividing cells that move. Okay. The other thing that I would like to do here is to that moving cell is an example of a structural compartmentalization of a cell. Okay. What I want to now is to explore the functional compartmentalization of a cell. Okay. So I want to proceed to make an exercise to create the base simulation that will be that will allow us to do this. Okay. Where you're now creating epithelia and you are dividing your cells into basal, lateral, and apical compartments. Okay. And they sit on top on an ECM layer. And then you can do this part. Because you're running out of time, I'll probably skip the model building part of this one and proceed directly to the choice of the correct contact energies. Okay. But let me just walk you through what's the idea. The idea is the following: is we have to create a multi plate, adjust cell areas, group cells together, right? So the group cell has assigned the same cluster ID for all the cells that are in the same Y position, X position, right? And then adjust the contact energy between the cells so they behave correctly. Okay, so well, we'll skip the model template. Okay, we call it tissue folding, Python XML. All these you get the apical lateral basal ECM cells. Okay, and then if you're making a model from scratch, make sure that you choose the contact and contact internal in your wizard. Okay, and then for this case, you choose volume flex, center of mass cell neighbors. And then what I want to do, okay, do this, and you get, and then when you just load, do the steps, you will get something that will not look like this. And part of this exercise is to make, to go to the, use the uniform initializer to make sure that you have a line of red cells on top, a line of green cells in the middle, a line of blue cells at the bottom, and then a line of gray cells here. Now we will skip this part. I want you to just get the code, okay? And then the way that you do this, if you want to have this kind of initial layout, you in the uniform initializer you can make multiple regions to define those different to make this arrangement, and then you make a first region that goes from X from for the just the apical sides, another region just for the lateral sides, another region just for the basal sides, and then another region just for the ECM cell type. Okay. Okay, so this should not be yeah, so X go from 0 to 250 because that's the size of our lattice, and Y goes just from 49 to 56. Well, the X is always from 0 to 250 because to span everything, and this just from here to here, the green just from 42 to 49, the blue from 42 to 35 to 42, and the gray, which is the ECM type, goes from 0 to 35, and then you get rid of those 3 layers, okay? And what I want to do, I don't want you to waste time going over all this model bin. I want you to go directly and grab the code 6.1.2b zip file, which already have all this implemented, okay? So you still can review the slides to see exactly how it's done, but I want you just to grab this file and let's start from here. Okay. So please download this file here on the student materials. Unzip, open on your Tweedit, plain Compartmental 3D, and let me know if you tell me what you see. Okay, Recon and Abhishek were able to load the code and run. Excellent, and you can see that things are not looking right. Okay. The code already has an adjustment for the keep the cell size. It does not adjust the ECM, right? And so the ECM cells quickly shrink. So what you have to, so if you just open the code, you may see something that is not correct. Okay. So what you have to do now, you have to do some work to first, let's make sure that everything is stable. So in the code that I sent you, we are defining ACM to be very big cells. So the first thing you need to do is make sure you got a stable object, right? So in the start function, there is should already be a code to add the volume of everything. So make sure that when you go over all the cell ECM, you assign a bigger value than what you had before, so it will not shrink. So that value should be, let me go back here, is 35 by 35, so we have to add this number square to the ECM cells. Let me open here on my tweeted, and then I can show you how the simulation looks like. Okay, so here you should have download the 6.1.2b file, okay, because everything's already set like this. And you see that here the plugin volume, you are assigning a volume for each one of the cells with 50, 50, 50. So that's why these cells here look okay. But the ECM, because you are assigning it to be big cells from 0 to 35, you need to change this value to be 35 squared. Okay, so 35 squared is 1, 2, 1, 2, 2, 5. Okay. So if you set this number here to this, then you're gonna have very big ECM cells. Does it work now?
I haven't added that self.reassign yet.
I didn't get what you said.
So in the main slide you have that self.reassign cluster ID cell2. I haven't done that yet.
It's not showing the cell type columns. I don't know. No, it doesn't matter. 50 and 49 are almost the same. You can change the cell colors to be whatever you want. It's just that because we made the ECM to be very big, we have to just find here, change this from 50 to 1225.
Yeah, now I will.
Now I got it? Okay. So
Yeah.
I know you got it. Kishore got it. Ali got it. Anderson, you probably download the wrong version of the file. It should be 6.1.2b with B at the end. We only have a few minutes left on this exercise. I want to make sure that everybody got at least at this stage here. Okay, so if you got this stage, you see that, okay, you don't really have a epithelium yet. Okay, so Susan, if you are there, then what you need to do is just set this number here to 1225, okay? And then you have the cells at the bottom be big. Okay, so change this line here. Okay, so if you got into this stage here, you see that they are not behaving epithelium, the cells are not linked together, okay? So what you have to do is now make sure that the cells on top of one another belong to the same cluster. And you have to do this by using a reassign cluster ID, okay? So how you can do this? What you have to do, you have to do a looper over one cell type. Let's say you make a loop over all the red cells, okay? Then inside that cell, you do a second loop over all the green cells, for example. And if they both have the same center of mass on the X position, then you do their cluster ID reassignment. And then you do another loop for all the blue cells. And if the X position matches, you reassign the cluster ID. Okay. So this is how you do in code-wise, okay? I don't want you guys to code this now because I already went over time. So I just want to follow up the slides and everything is already solved. So you can download the zip files, you can see the solved problem, okay? So the way it solves the following, you go, for example, all the cells are this apical, and then once you grab the cell, you do a second cell loop. Then you have to assign this dummy variable a different number, cell 2, of all the cells lateral. Check if the center of mass are the same, and then you make a reassignment. Okay. You say that this cell 2 now have the same cluster D of the cell that was the top. You do the same for the basal. And what you have, you end up with now all those cells have the same, belong to the same cluster. Okay. But if you just do that in one simulation, they're gonna still not behave correctly because they're kind of flopping one another. They're still behaving as a single cell, right? It's just now the boundaries are a little bit different, but still wrong. So the way you have to do this now, once you have this implemented, you have to go into the contact energies and make a choice of what the contact energy should be. Okay, if you want to have a stable epithelium like this, how does the energies between the different cell types should be, what the energies internally should be, okay, and what the external energies should be. Choose those energies values here. So did anyone was successful in solving this problem here? It seems that Kishore was able to do it. So, but everybody can see the chat and Vikram got it. One thing that I can tell you is that sometimes you cannot tell if the boundary you see is from the cell or from the or is a cell boundary or is a cell cluster boundary. Okay, so there's a way to fix this. Let me just, I guess, if you're seeing this video of the crawling cell, let me make a zoom, right? You see that you have boundaries everywhere, but if you go to the visualization, you also can have an option for cell borders. Okay, and if you click on the tools and go to cell colors, you can choose a color for the cell boundaries and a color for the cluster boundaries. So, for example, I may want to make my cluster boundary to be magenta, okay, and my cell boundary to be, let's say black, okay. I hit OK, and what you see now is that not sure if it's really visible. Maybe I should choose another. But you should see that is magenta here around the cells. So this maybe a bad example because I don't have a cluster yet. Let me open exactly this example. So here now, I set my cell boundary colors to be white and my cluster boundary to be black. Maybe I can choose another one. Let me choose something that's very visible, yellow, okay? Hit OK. And then you can see, okay, the yellow boundaries are the cluster boundaries and the white boundaries are the boundaries within the cluster, within the cells, so you can clearly tell who belongs to whom. What I like to do when I'm using cell compartments is to not have the boundary between the cells, only have the boundary between the clusters. So I usually choose this out, and then you see here that you don't have a boundary between all the red, the green, the blue compartments, because that indicates right there they belong to the same cluster. And I think this is a better visualization. And then I also like to have the cluster boundary to be black, so you can have the same thing on the screen. I like this choice, so where you see a boundary is between, so you can clearly see that this belongs to the same cluster, and those two here belong to the different clusters. Or if you want to highlight that even more, you can make the cluster boundary color to be white. Okay, and that's another choice that highlights, okay, these are the cells, these are the compartments, and this is the cluster. Okay, so but that's a minor thing, just to show that you have this kind of choices. So let's go back to the exercise. So how you go about making a stable epithelium, you have to make a choice about which ends should be high, ends should be low. Let's start with the contact internal, okay? So we want the internal compartments to be cohesive. So you probably want the apical, lateral, apical, basal, and basolateral to have low contact energies. So then they're going to stay as a whole. Okay. If you want to enforce that the apical is say once on the opposite side of the basal compartment, you may increase the apical and basal compartment to be a little bit higher. Okay. So they're going to be the opposite sides of your cluster. Okay. Again, there are no cells with the median or ECM, so we can ignore all those values here on the top, on the lateral. Okay. In this case, you also do not have repeated domains. You don't have two apical sites or two lateral sites or two basal sites in the cell. So that means that everything that lies on this diagonal here can also be ignored. In some cases, you may have two compartments at the same time in your cell. For example, if you want to model a cell with two nuclei, that may happen, okay? Either because of disease or because you do have a nucleated cell. That happens in some developmental processes. You have two nuclei, then you have to actually specify the energy here. But in this case, that's all you have to do in the internal contact energies. Now let's go to the other side, the external contact energies. Okay. You want to have coupling between the apical, lateral, and basal. So you want the cells to not slide one another from top to bottom, but you want to have they making a line along the horizontal, like. So you want to probably have a low contact energy between neighboring apical sites, neighboring lateral sites, and neighboring basal sites. If you already know that all your adhesion junctions are on the lateral side, you may make this lower and those two a little bit higher. But in principle, you want to make this low, but not as low as the internal. So it's going to be a little bit higher than the internal ones. Okay. So I hope this makes sense. So let's now proceed to what should be the cross, right? Apical to this lateral, apical to this basal, or basal to this lateral, and so what about this cross terms here? So you probably do not want the basal to be touching the apical side of the other cell and vice versa, or even touch the lateral to be touching the apical side or the base of the other cells. We want to enforced alignment. So in this case, you may make the case, okay, that these energies should be high, okay? So that will, this together with this will enforce the alignment of our epithelium. And epithelium cells are aligned with one another, okay? So you choose them to be higher. Now, the next thing you want to see, okay, how should it be the contact of those cell compartments with the ECM and the medium? Because you want the apical, let's start with the apical side. You want the apical sides to see the medium and to not see the ECM. So what you do, you have a low contact energy of the apical side with the medium and a high contact energy of the apical side with the ECM on the bottom. So you want this value to be high. You can choose the same principle for the basal side. You want it to have a high energy with the medium and a low energy with the ACM. So we make this high and this low. Again, you don't want to get, in principle, you don't want this contact energy to be lower than the internal contact energy. So it always should be, your first choice should be, even though you choose to be low here, it has to be a little bit higher than the internal ones. Okay. Now, next thing you have to do, okay, what about the lateral side? How it should interact with the medium and the ACM? You usually don't want this to interact at all, so you also keep those two values high. And finally, the contact of ECM with other ECM cells, you should be treating as a continuum, so you can just set this to be 0, okay? And the ECM should never see the medium, okay? So you can also set this the last hint to be high. And once you make this kind of choices, and you see that's only qualitatively, you have to think about the behaviors. You run your simulation, and then you should have a stable epithelium. So if as long as you follow this kind of hierarchy here, you should get it. But just to show an example, if I make this kind of choices, it also works, and you should get something that looks like this. And this code with this kind of implementation can be found on file 6.1.2D zip, and this is going to be from the basis for you to then do the epithelial folding that you're going to learn on module 7.4. But before you can do that with epithelium, with cell compartments, you first have to learn how to add links in the cells themselves without the compartments. And that is the theme of this module. So before I proceed, do you guys have any questions about this part?
So in this case, only the cells in the ECM are dividing, right?
No, the ECM is not dividing. It's just because I set the contact energy to be
Okay.
0, they mix.
Okay.
The whole question about forces is actually the topic of this module that we are now with the 6.2. So we'll be talking about forces right now. Okay. Well, the contact energy between cells and ECM are set here. It's going to be this. That was your question, is that here the apical side does not like to touch the ECM, so it's high. The lateral side also doesn't like it, so it's also quite high. But the basal sides do like the being contact ECM, so this is low, 5.
I see. Okay. Thank you.
Okay. So if you want to know how to do this, add those forces within the apical side that drives a foreign formation, we have to go about how